to acetyl-CoA. Acetyl-CoA is 2 carbons, it will attach 1 more carbon, and now you end up with 3 carbons. And the name of that molecule is called malonyl-CoA. And there's my acetyl-CoA carboxylase. I need ATP and carbon dioxide for that to end up with a 3-carbon molecule called malonyl-CoA. So, I now have, in the cytoplasm, I have acetyl-CoA which came from the mitochondria as citrate. Okay, fine. I've taken some of that acetyl-CoA and I've converted it into malonyl-CoA. So I've got some acetyl-CoA and some malonyl-CoA. In other words, I've got a 2-carbon molecule and I've got some that is in the form of a 3-carbon molecule. You got this? Now, I can use those 2 things, the 2-carbon molecule and the 3-carbon molecule, all right? And I can now make a fatty acid chain. And the body makes a saturated fat called palmitoyl-CoA or palmitate. Okay? For that, I need another enzyme called fatty acid synthase. Fatty acid synthase is actually 5 enzymes all in one. But I'll come to that one later. Let's just look at this acetyl-CoA quickly because it is the regulated step of fat synthesis. And the first step of fatty acid, acetyl-CoA carboxylase, the first step of the reaction, there's the enzyme. It's made up of 3 parts. I have a biotin holding part, then I attach the carbon dioxide to the biotin arm part, then transfer the whole thing over the transcarboxylase part, and that will transfer the little green thing onto the malonyl, and there's my 3-carbon malonyl. This will now switch back to this form to pick up the next carbon dioxide. So that is how acetyl-CoA will attach. Acetyl-CoA carboxylase will attach a carbon dioxide to the acetyl-CoA. Good. So in the cytoplasm, I have some acetyl-CoA, I have some malonyl-CoA. Are we okay? Now what do I need to do? I need to activate both of these. So there we are. I activate both of them with a thing called ACP. Okay? It's called acyl carrier protein because I cannot carry out any chemical reactions on acetyl alone or malonyl alone. It's got to be attached to my friend ACP, acyl carrier protein. So now I attach those. So ACP acetyl, ACP malonyl. Now I am ready. For who? FAS. Fatty acyl synthase. Well, fatty acyl synthase, again, I told you it's about 5 enzymes all in one, working all together. It catalyzes a stepwise synthesis of a C16 fatty acid chain. You require NADPH, and it goes through a cycle. Each cycle, it attaches 2 carbons at a time until I get to C16. Obviously, that means there are, if I start off with 1 acetyl-CoA, there are 7 cycles, okay, which will give me the 16. All right? Now, let's look at this attachment process. So I start off with an acetyl-CoA all the way up on the top. Can you see the acetyl-CoA? Can you see the malonyl-CoA? The malonyl is 3 carbons, the acetyl is 2. If I add them both up through a condensation reaction, I should actually get 5. But what happens is, I release 1. So now, can you see the carbon dioxide coming out? And now I've got a 4-carbon, condensed 4-carbon molecule. Don't worry about its name. Okay? Then what do I do? That's the condensation part. I bring it together. That's what condensation means. Then I carry out a reduction reaction. In other words, I release a pair of electrons. Okay? I release a pair of electrons. I add a pair of electrons. I add a pair of electrons and hydrogen, and now I come up with this molecule down here. Then I remove a molecule of water, dehydration reaction. And again, I add another pair of electrons, okay, a reduction reaction. And I end up with a four carbon chain, which I will then take all the way up, back up to this blue position. Can you see it? Can you see this blue position up here? I take it all the way back up. So I will start with 4, and then I've got another malonyl ACP. And I now start again. So 4 plus 3 is 7, but I release 1 carbon, and now I will have 6. I carry out a reduction, dehydration reduction, and then I got 6. You follow? And that's what happens during fatty acid synthesis. So you have a condensation reaction, a reduction reaction, a dehydration reaction, and then lastly another reduction reaction. Do you need to know the chemical figures? No, you don't need to know the chemical figures. Do you need to know the sequence of the reaction? Yes, that's important. Do you need to know the name? No, that's not important. Now, how do you regulate fatty acid synthesis? Well, there we go. I make citrate, which becomes acetyl-CoA. Obviously, citrate made in the mitochondria goes out into the cytoplasm and becomes acetyl-CoA. In the cytoplasm, ATP citrate lyase would chop the citrate into OAA, or oxaloacetate, and acetyl-CoA. Now, that particular enzyme is stimulated, ATP citrate lyase is stimulated by insulin. Okay? Which kind of makes sense because in an energy-rich state where there's a lot of ATP, NADH, you've got full stores of glycogen and so on and so forth, your insulin levels are high, therefore you stimulate this enzyme to carry out fatty acid synthesis. Then your acetyl-CoA will be converted into malonyl-CoA by acetyl-CoA carboxylase. Again, this particular enzyme can be stimulated, all right, by citrate, like an allosteric activator. Good. Acetyl-CoA carboxylase can be inhibited by glucagon, epinephrine, can be inhibited by palmitoyl-CoA, in other words, end product inhibition. All right? Good. The next thing about this enzyme is it works through covalent modification as well. So when it is phosphorylated, it doesn't work. All right? When it is phosphorylated. When it is dephosphorylated through a phosphatase. When you phosphorylate, you need a kinase. When you dephosphorylate, you need a phosphatase. When it is in a dephosphorylated state, it works. So anything that will dephosphorylate it will make the enzyme work. And if you look at the bottom, okay, high energy state, in other words, more ATP than AMP, insulin, activation of the phosphatase, all right, will make this enzyme work. Anything that will phosphorylate it will stop the enzyme from working. And what is that? More AMP. More AMP than ATP, okay? Increase in AMP, or AMP kinase, which will activate protein kinase A, which will then phosphorylate the enzyme, and things like glucagon and epinephrine. Good. One more thing about this enzyme. It works better when it's in a group. Even though I have individual enzymes here, it works better when it's like kind of polymerized. And who stimulates the polymerization of the enzyme? Citrate. Helps it to become, okay, group mentality if you want. Okay, so much for this enzyme. Next thing. So I've made my fatty acid. Where is it? It's in the cytoplasm. All right? Now, I can attach my fatty acid to a triglycerol. I'm just going to skip a couple of slides. To make my triglyceride, I've made my fatty acid. To make my triglyceride, I need one more thing. I've got my fatty acid. Hopefully I have 3 of them. I need one more thing, and I need glycerol. Where do I get glycerol? There we go. Again, in an energy-rich state, I convert some of my pyruvate into DHAP. There's DHAP here. And then I convert that into glycerol 3-phosphate. Together with 3 fatty acids, I can have triglycerides. So I make these triglycerides, then I cannot put them into blood. Why? Because it's a fat. Okay? 90% of plasma is water. That thing's not going to go anywhere. It's just going to get stuck on the endothelium. So I take the triglyceride, okay, and I put it into the middle of a phospholipid layer kind of thing, single layer on my cell. I put a chunk of protein here and there, okay, the apoproteins, and together with the triglyceride, I put some cholesterol inside. Now, I can put that into blood. Why? Because the outside phospholipid and protein will protect that thing from coming out. Because in the blood, I cannot have bile salts. You understand? Because that's exactly what bile salts do in the intestine. The same thing. But here, what I do is I turn it into a lipoprotein. Okay? And that name of that lipoprotein, if this is happening in the intestine, in the enterocyte, is called chylomicron. If it is happening in the liver, it's called VLDL. All right? Good. Now, that means in our blood right now, we have these these chylomicrons flowing. Now, what happens near the tissue, near muscle, near the cardiac muscle, near the liver, near the adipose tissue? What's happening? Well, this is what's happening. There's that triglyceride inside the lipoprotein. It comes close to the endothelium. There's the endothelium. This is the cell. And sticking out of the endothelium layer is an enzyme called lipoprotein lipase. That lipoprotein lipase will first break down the triglyceride into the fatty acids and the glycerol, and those will then be absorbed individually through the endothelium and then into the adipose tissue. Now, I have another version of this enzyme that breaks down triglycerides, and the other version of it is inside the adipose tissue. It's called hormone-sensitive lipase. And it breaks down fatty acids inside the cell. So this guy breaks down the triglycerides outside the cell. This one breaks down triglycerides inside the cell. Obviously, this one breaks down the triglycerides so you can allow stuff to go in. This one breaks down triglyceride inside the cell so you allow the stuff to go out. So they obviously cannot work together, which kind of makes sense. That is why insulin stimulates this guy to allow the fatty acids and the glycerol to go in, whereas insulin will inhibit this guy because you cannot be pushing out and pushing in at the same time. So insulin inhibits this guy. How? By removing the phosphate. On the other hand, if you are hypoglycemic, your insulin levels are very gone, but your glucagon levels are high. Look at what happens. Epinephrine or glucagon binding to its receptor will activate protein kinase A. Protein kinase A will phosphorylate this thing, and now you can break down your triglycerides into fatty acids. Okay? Are we all right so far? Good. So, we need to regulate not just the fatty acid synthesis, but we also need to regulate glycerol synthesis. And I told you in class that insulin in people with type 2 diabetics, they do not have less insulin. If anything, they have more insulin than they need. But they are insulin resistant. The insulin is not working. The insulin, for some reason or not, cannot tell the GLUT4 transporters, which are in the form of vesicles inside the cell, they cannot tell them, you know what, please go onto the membrane and let the glucose come in. They can't do that. One of the theories goes something like this. In people who have type 2 diabetes, they also have dyslipidemia. In other words, they have high fatty acid content inside the cell. The fatty acid interferes with the secondary signaling inside the cell. So, what do? You as doctors would prescribe a drug called a glitazone, TZD. What does TZD do? It stimulates. Can you see down here at the bottom? Yes? It stimulates PEP carboxykinase. That means you convert pyruvate to DHAP to glycerol. Now, inside the cell, the glycerol can join up with the fatty acid to give me a triglyceride. That means you remove, you tie up the triglycerides, and you remove the fatty acids away from the signaling pathway, and now the insulin can work a little bit better. Okay? And that's what this is saying. All right? So by activating PEP carboxykinase using glitazones, we actually make insulin less resistant. You follow the logic here. Okay? Good. Let's try the first question. Now, I have some questions that is just, I put it in there just to see whether you can recall the content information. Sometimes it's more than one step in the content information. Other times, okay, it is a clinical scenario. So, let's do a couple of questions. The first question. Choose the one best answer that most accurately describes some properties of acetyl-CoA carboxylase. Now, first one. Would you think it requires or it doesn't require biotin? Yes, it has a three-part thing. Would you think it requires thymine or it doesn't require thymine? No. I said nothing about thymine. This is mine. I wonder who drank my tea. Oh no, wrong cup. Yes. So it doesn't require thymine. So the answer is either A, B, and it cannot be E and F. Yes? Okay, let's go to the next one. Where in the cell will you find the acetyl-CoA carboxylase? Obviously, the reactions are in the cytoplasm. So it can be B, D, it can't be E because we already, it can't be F. All right? Again, can you change the acetyl-CoA carboxylase in some way? Yes, you can. You can phosphorylate it, you can polymerize it, you can make it into a chain. Yes, you can. Who does that? Citrate. You notice the only answer it could be is? Can somebody explain the last column, AMP activated protein kinase for me? Why will that affect the acetyl-CoA carboxylase? Yes, Ariz, you had your hand up.
So AMP is a signal of low energy inside the cell.
Yeah?
You would only synthesize fatty acid in high energy states.
Yes.
High ATP. You would need to, so AMP inactivates fatty acid so you can do gluconeogenesis.
Yeah?
Also, it's also in a protein kinase phosphorylates and it's inactivated by phosphorylation.
Yeah, but look at it. It says AMP activated protein kinase.
AMP is a low energy signal.
Just 1 minute. Yes, 1 minute. And in a phosphorylated state, it works or it doesn't work? It doesn't work. Okay? All right? So you understood what I just explained to you in the presentation. So you were right. Who said B at the very beginning? Let's try number 2. That's the answer. I noticed somebody up there is putting me on YouTube. I don't know why. Okay? But that's the answer. Okay? Again, citrate is an allosteric. You know what the word allosteric means. You've done the enzyme lecture. Yeah. Okay? Is an allosteric activator of the enzyme. Okay? And the enzyme is inhibited when it is phosphorylated, as you rightfully pointed out. Second question. The synthesis of fatty acids from glucose in the liver is best described by which of the following? The pathway occurs solely in the mitochondria? No. Wrong. Okay? It says fatty acids from glucose. It takes place in the cytoplasm. Yes? Yes, there is one part that's required in the mitochondria, the pyruvate to citrate going out. But it's not solely in the mitochondria. It requires a covalently bound derivative of pantothenic acid. Do you still remember what pantothenic acid is?
B5?
Yeah?
Vitamin B5?
Yeah? Well, you find out. It requires NADPH derived solely. You know what this word solely means? Only from the pentose phosphate pathway. True? No. No. Because you can make the NADPH when you convert oxaloacetate back to pyruvate in the cytoplasm. The pathway is primarily regulated by isocitrate.
Yes.
No. Citrate, yes, but not isocitrate. Next one. The pathway does not utilize a carboxylation reaction. It does. Because the only way you can convert acetyl-CoA to malonyl is through carboxylation. So the only answer is B. It requires a covalently bound derivative of pantothenic acid. Now, I don't know if I told you this or not, but there we go. And that's the answer. Because anytime you attach a carbon dioxide, you require biotin. Can you see the answer? Biotin is required for the conversion of pyruvate to oxaloacetate. You got this? Which combines with acetyl-CoA to form citrate. Biotin is required in the acetyl-CoA carboxylase reaction. All right? Then, 2 lines down, pantothenic acid is a covalently bound to the fatty acid synthase enzyme. Okay? To the fatty acid synthase enzyme as part of the phosphopantothenyl residue. Okay, next question. Now this is the one where you have to look at various things. Okay, which of the following best describes the synthesis of triglycerides in adipose tissue? All right? Now, in adipose tissue, in other words, the first column says source of the fatty acid. In other words, is the fatty acid coming from the enterocyte or is it coming from the liver to the adipose tissue? Both of them. So it be VLDL from liver, chylomicrons from enterocyte. Got that? So the answer has to be C and D. Forget everything else. Are we okay so far? Good. Next one. What is the source of the backbone? Means the carbon that attaches the fatty acids. Yes? The glycerol. That's exactly what I mean. But before I make the glycerol, I need exactly DHAP. So, okay, I'm still C and D. Does it require coenzyme A? Yes, it does. It requires coenzyme A. I'm still C and D. Okay? Does it require lipoprotein lipase to make? No. I'm still C and D. Now, how do I separate C and D? Huh? Well, there we go. You're right. It requires 3. It doesn't require 2 or 1. Okay? And again, that would be the no. All right? And there we go. You got the picture? Okay, there we are. Okay? Lipoprotein lipase is not required. Third line down, please. Glucose is converted to DHAP, which then is converted to glycerol 3-phosphate. After inorganic phosphate is released from the resultant diacyl glycerol react with another fatty acid to give you triacyl glycerol. Okay? Good. Next question. Which of the following best describes fasting conditions? Now, you're sitting in the room. I'm going to tell you now, I'm going to put a question like this in the exam. Okay? So, let's have a look. Fasting conditions. So, activity of hormone-sensitive lipase. You sure it will be active? Yes, okay, fine, all right. So I'm breaking down inside the cell and I'm releasing into the blood in a fasting condition, yes? The fate of the glycerol. What happens to the glycerol in this process? Sorry, active is B, D, and F, yes? So, now I've got gluconeogenesis. We all agree on that one? Good. Now I've got D and F. VLDL production. VLDL production. VLDL production, yes? I've got D and I've got F. I'm only left with D and F now. Both are low. Yes? Good. Alright? Low. Next one. Modification of acetyl-CoA carboxylase. Phosphorylated, both of them, yes? Because I don't want to be synthesizing while I'm breaking down. So both are phosphorylated. Lastly, ketone body production. Yes? Because we got a no in D, we got a yes in F. Yes. Fasting conditions. Hypoglycemic fasting conditions. Okay? You will produce ketone bodies. So, again, there's the explanation. I'll just read the last bit. Glycerol is converted to glucose by gluconeogenesis. Fatty acids are oxidized to produce ketone bodies. Alright? Because the brain and the muscle and the heart can preferentially use ketone bodies. What's the name of the enzyme? Thio, yes, thiopherase. Okay? Next one. Now there are 4 questions here. I'll quickly read this. It says a molecule of palmitate or palmitic acid, which is the fatty acid our body makes, attached to carbon number 1 of the glycerol molecule of a triacyl glycerol, is ingested and digested. It passes into the blood, is stored in the fat cell, and ultimately is oxidized into carbon dioxide and water in a muscle cell. Choose the molecular complex in the blood in which the palmitate residue is carried from the first site to the second in each of the 4 questions that follow. So, first question, how does the palmitate travel from the lumen of the gut to the surface of the gut cell? Yes? D. D. From the intestine lumen to the cell. D. The micelle formation because of bile salts. Next one. From the gut epithelial cell to the blood.
B.
Okay, yes? Chylomicrons. Next one. From the intestine through the blood to a fat cell. From the intestine through the blood to a fat cell. Yes? Again, chylomicron. Yes? Lastly, from a fat cell to a muscle cell. Yes? Albumin. And there's the answers. DBBC. Next question. A 4-month-old child. What does that tell you immediately? Good, inborn error. Immediately. Don't even read the rest of the question. Inborn error. Missing an enzyme. Something is missing. Okay. A 4-month-old child shows extreme tiredness, irritable moods, poor appetite, fasting hypoglycemia. Fasting hypoglycemia associated with vomiting and muscle weakness. Blood work shows elevated levels of free fatty acids. All right? But low levels of acyl carnitine. A muscle biopsy shows significant levels of fatty acid infiltration in the cytoplasm of the muscle. The most likely molecular defect in this child is which of the following? Yes. D? Yeah? HNG-CoA synthase? Who said E? Well, this child is suffering from an inability to transport fatty acids into the mitochondria. Now, it could be CPT1, CPT2, or it could be one more thing. What is the one more thing? Carnitine. Acyl carnitine. You're missing carnitine. In other words, the transporters CPT1 and CPT2 are working okay. So you could be missing the actual carnitine. All right? And this child is missing that. And there's the answer. Okay? So you're missing carnitine as a transporter. And there we go. The child has symptoms of primary carnitine deficiency. Okay? And without the carnitine, you cannot transport the fatty acids, okay, from the blood into the liver. So they all stay in the blood or into the muscle, so that they can be oxidized off. Inability to utilize fatty acids for energy gives rise to muscle weakness. Accumulation of fatty acids can occur within the muscle tissue. All right? Now you are going to ask me, if I don't have the carnitine, how is the fatty acid getting into the muscle tissue? And I'm going to quickly move this thing back to the question, because I don't want you to read the answer. Can somebody tell me what accumulation is taking place in the muscle? If I don't have the carnitine, I can't get the fatty acid in. What fatty acid is accumulating in the muscle?
Coenzyme fatty acid. acyl-CoA maybe.
Short chain fatty acids. They do not require a transporter because they are water soluble and they can go through. And those are the ones that are accumulating inside the, yes? Inside the muscle.
Okay, you can read this on your own. I think that's the last